Mm.
Okay, hi everyone. So let's get started. Welcome to today's webinar on accelerating antibody discovery. We have a full agenda and exciting topics today, but before I get started, I just want to remind you this webinar will be recorded and you'll receive it after today's session. And we also have a special bonus for attendees that stay to the end, so be sure to stick around for that. For me, my name is Monica. I am the head of go-to-market at my laboratories, and let us get started with today's session. So to start, I wanted to start off with a poll. So let me just launch it right now. And the question is, what type of antibody discovery approach are you currently using in your research or work? And you have a range of in vitro, in vivo, hybrid, in silico, or maybe you're not doing any antibody discovery just yet. So I'll give everyone 20 more seconds to answer. So it seems like most people are doing in vitro. In vivo is also pretty popular, and a lot of people are also doing both. Great. Okay, so let me go back to sharing the slides.
Yeah, yeah, I'm sharing.
Software
Yeah.
does have capabilities for in vivo. We can cover everything today, so we'll just be focusing on in vitro in today's discussion. If you're interested in vivo, we'll have a separate dedicated section next month, or just shoot me a message if you want to learn more. And also feel free to drop any questions you have in the chat, and we'll get to them at the Q&A for today. So to set the scene, in vitro discovery uses display technologies to find high affinity leads. And how that works is we start with a high diversity initial screening library. Then we perform rounds of panning with phage or yeast to select the high affinity leads. And then these leads are ready for deeper characterization and selection. So now that we walk through this cycle, the next question is, how do we make it smarter? NGS, on top of other functional assays, allows us to go deeper. So for traditional methods like single sequencing, you're actually just looking at the tip of the iceberg to a tiny fraction of all clones. And that means you're likely just picking the most dominant clones, not necessarily the highest affinity binders. In doing so, you're missing what is underneath the water. So NGS, on the other hand, gives you a complete view of the repertoire, which means that we can detect rare, high affinity variants that wouldn't show up in traditional screening. The advantage here is that you can map full families and lineages. You can identify backup candidates if your lead that you picked failed, and you can also rescue high affinity binders that are just growing slower. So to summarize, single sequencing allows for just hundreds of clones, while NGS will give you breadth and depth to millions of sequences. So given everything we just discussed, if NGS is such a gold mine, then why doesn't everyone do it? And that's because the analysis step has become a big bottleneck. Advances in the wet lab has become a lot faster and a lot more efficient. But once you generate these massive data sets from NGS, the real challenge begins. Analysis is slow, it's fragmented, it's code heavy, the things that we hear very often, and it requires specialized expertise, and most teams don't really have that bioinformatics resource or computational power. That slows the entire workflow down, and that's exactly what we're going to be addressing today. And let's talk about exactly what we're trying to get out of the analysis. So researchers need the right tools to equip them to prioritize the right candidates. From the outputs of such big data sets, what we need to know is which sequences have high affinity to the target antigen, which clones are expanding across treatments or patient groups, and can we predict developability, directly from the antibody sequence to select the top candidates. If you have these questions too, you are in the right place. So for today's agenda, I'll pass it over to Stan, the CEO of My Laboratories, who will give you an introduction to the company. Then he'll go over today's case study and give a live demo with our head of customer support, Mark Israelson. And then at the end of that, we'll save time for questions. And just a reminder, we'll be revealing a special bonus offer at the end just for attendees of this webinar. So Stan, I will now pass it over to you.
Thank you very much, Monica, for the introduction. Yeah, so what we will be doing today, we'll be doing a live demo with a case study of VHH antibody discovery with phage displays. So to start with, let me introduce ourselves, who we are, what's our company, and why we are doing this. Yeah. So historically, our background of almost all of our team members, like Mark here, we work with Mark for more than 10 years, or our chief technology officer, we worked with him for 15 years in academia. So we did a lot of research in academia, and one of the tools that we created in the immune sequencing space is called MyXCR. So that's a very well-known tool and widely used. So we first released the first version of MyXCR in 2013. It was called MyTCR for T-cell receptors. Then in 2015, we released MyXCR, where X stands for T or B-cell receptors. Yeah, it was published in Nature Methods, Nature Biotechnology, and today it's the most widely used tool for BCR and TCR NGS sequencing data analysis. Lots of citations, lots of data processed, and actually we put our hands on all tons of that data, especially Mark. So I noticed some of the attendees of this webinar had already a lot of communications with Mark regarding various support questions, how to run their BCR analysis on this protocol or that protocol, et cetera, et cetera. Just one number, over the past 7 years, more than 10 million of samples were processed with MyXCR. And we appreciate, of course, many people today are joining from the industry, and we are a company, so we provide our solutions for leading biopharma and biotech and pharma companies. 8 out of 10 big pharma companies use our software. So we have more than 1,000 of organizations using MyXCR and other tools today, Platforma, on the daily basis, including government agencies, research institutions, and universities, and we work with them a lot with some initiatives with FDA or CDC or National Health. Yeah, and with this introduction to give us some credibility why we are doing the webinar about antibody discovery, yeah, I'm switching to what we are showing today. That's the platform, the software that we were building for past couple of years, actually 5 years. It's called Platforma, and it provides a unified discovery workspace to empower biologists. Today, specifically, we are focusing on the antibody discovery. So I'm talking about biologists who are doing antibody discovery with NGS data, to drive their analysis and get results from FASTQ files, from the raw sequencing data, assay data, to the final down selection of lead candidates. It is a zero-code platform with intuitive graphical user interface, which speaks biological and language. So to run the pipeline, to get the results, you don't need Python or R scripts. Everything is integrated, functional data, NGS data, the analysis. It's very transparent. So the computational biologists who are always validating the platform and how it works, like, perfectly knows all the tools and all the pipelines and algorithms that are lying under the hood of what we will be showing, and they actually open sourced and built on the gold standards of the industry. And what we will be doing, we will be showing how you can integrate all the sources of the data to get like really deep biological results yourself. Pretty fast, assess developability risk, integrate functional assay data, and build the diverse panel of antibody lead candidates. The case study that we will be working with you today is the well-known publication. It's pretty well cited, published in Nature Communications. So the case study is a rapid isolation of nanobodies against SARS coronavirus. The goal of this publication was to isolate potent nanobodies to neutralize spike protein during the early pandemic response. So very relevant stuff. And the methodology used in the publication is pretty straightforward and pretty common for the in vitro antibody discovery. So the source of the screening library, the highly diverse library, was Tyson. I don't know, but I hope Tyson is still alive and doing well. So it's alpaca, and he was this alpaca. It was immunized with the SARS-CoV-2 S1 FC and RBD over the 60-day schedule. Then the PBMC was isolated, and 2 panning rounds after the screening library were performed with phage display. And apart from just picking the top with binders with Sanger and Say methodologists, scientists behind this publication also collected the NGS data. So we have FASTQ files, and we're now able to look on the FASTQ files and find the exact TY1 nanobody that was reported as the discovery as a neutralizing antibody. And also look wider and see whether we have something else in that data. And yeah, so that's actually what we will be doing. The authors of the publication, they found the lead candidate, one lead candidate, TY1. We're going to see whether there is something more interesting. This TY1 was validated. It has high affinity. It is a really good candidate with the clear NGS support. In the study, the NGS dataset was used as a support for the discovery, while we are going to slightly change it and use NGS data as a driver for our discovery that we will further validate with the functional assay data. So with this setup, I will switch to the software, to the live demonstration. Please don't forget to ask your questions in the Q&A session. We will aim to reproduce the results of this study within, well, I'm not saying minutes, but at least maybe 20 minutes. That's our challenge for today. Meanwhile, Monica is launching the next poll in the chat with the question on which tools you use for antibody discovery in the software side. So I switch my screen to the Platforma, to the software that I will be showing you today. There are 2 components of the software. One component is the graphical user interface that you install on your personal computer, and you can install it on your Linux. I'm joking. Wet lab scientists don't have Linux. Of course, Windows, Mac, or yes, also Linux. That's one component. And when you install it and launch, the first screen that you will see is the following: where to run the analysis. There are a couple of options. I will be exploring the cloud option with this option, the backend, the server, where the data will be processed with the heavy bioinformatic tools as located in my laboratory's cloud. But if you run Platforma in your server on premise, then you can install the server on premise and connect to it. Or if you don't use like a huge terabytes data sets, that you can even run the whole analysis on your personal computer with a built-in mod. So I'm connecting to the cloud, and what you will see when you run Platforma is obviously I have some projects here. In your case, it will be empty, or if you use our cloud, then you will have some demo projects already pre-processed here. Project that we are going to explore is a TY webinar. So I'm just showing you right now what we will achieve during our live demo. So we will start with the raw FASTQ files from the very beginning to extracting, annotating antibodies, clustering antibodies into families, and doing enrichment analysis to find exactly the clusters and antibodies that are enriched through rounds of selection, exploring the entire space of our antibodies to see, I have some filters here, to see them and the enrichment factors right on the interactive UMAP plot, and doing a lot of other things toward the final goal, building the list of high affinity candidates. So that's what we will be doing. Let's start going step by step and aim to get this result and find our TY1 nanobody as well as our other cousins of this nanobody from this data. So I'm starting with creating a new project. So let's call it webinar. Oops, webinar live. And the some of the things I will be covering really fast, really quick. They are not very interesting, but the entry point of any project is important, the raw data. In our case, it's FASTQ files, and we can connect the remote storage or your system where you have the raw data coming from your NGS, and it will be available at the remote tab where we can select, let's say I have some FASTQ files. Those are the FASTQ files corresponding to this specific study. You can select all the FASTQ files and bring them into the project. By the way, you already noticed that on the left, we have a side panel, a left panel where we have those blocks. We call them blocks. Oh, they are like a biological applications designed for various biological needs. And the first one, samples and data, is the place where we are defining the data and ingesting the data into the project. So in our case, it's FASTQ files. Let's call this data set VHH. There are various capabilities how we can import the data, and we support all types of data, not even necessarily FASTQ files. It may be FASTA files, or just clonotype tables, or whatever. So the platform is really flexible. And let's create this data set. So we have 4 samples. For every sample, we have a pair of NGS FASTQ files, read 1 and read 2. And before we proceed downstream to getting the analysis done, the crucial step is to attach the metadata. And in our case, for in vitro antibody discovery, the most critical metadata is the enrichment round, which samples belong to screening library and which samples belong to which pinning rounds, which can be done here in the metadata section with import metadata button. And here we can import just any Excel file or TSV, CSV file with only one requirement. This Excel sheet must have a sample name column, and other columns may have, well, whatever metadata you have. In our case, the most critical one and obligatory for real downstream selection is enrichment round. You may call it pinning round, enrichment round, selection, or whatever, with the values of the actual enrichment round. So I click Run button to make the import happening, and now we are building our down selection starting from the FASTQ files. The first step of the pipeline is to annotate our raw sequences and build the antibody clonotypes. I clicked that block, and here you see a set of applications or blocks that we have for various biological purposes. Specifically to extract, to annotate the clonotypes, we have a set of apps, a set of blocks based on MyXCR tool. The one that I'm going to use today is MyXCR clonotyping, although we have options for, let's say, for SCFE, for single chain variable fragments, or amplicon alignment for synthetic libraries. Here it's alpaca, so I'm using just MyXCR clonotyping. Let's add it to the project, and the settings are pretty straightforward. We are selecting the data set. We have only one in our project, the VHH. And then there are 2 options how we tell MyXCR what are the library preparation, like configuration of our NGS library. What's the library preparation protocol? If we use a commercial vendor for chemistry, for library preparation, then we can select the preset from the list of available presets. Or if we use a custom library preparation protocol, which is a frequent case, Mark, tell me how many people doing antibody discovery use custom library preparation protocols and how many use the vendors based on your tremendous amount of support.
I think that the majority of people, especially if we're talking about not academia, they of course use their in-house protocols. So like usually it's somehow like tuned to their specific needs, and we help them with the file, with the YAML file, with all of the settings that they need. But in academia, of course, a lot of times the like regular kits are used.
And in case of this specific study, although this is academic study, the custom library preparation protocol with custom primers was used. So there is an option to specify the parameters, put the parameters of this library in the file. And we have full documentation of this actually. So, and also we have support, and then Mark helps a lot to assemble this file. So I'm selecting this preset for the library preparation protocol used in this study here. Yeah, what's also important, since MyXR clone typing is the generic block for TCR and BCR clone typing analysis, the default receptors include everything. Of course, in our case, it's alpaca antibodies. We are selecting only immunoglobulin heavy. And then we click run. So it will actually start the analysis. It will run on the back end where Platforma is running. It will run the real MyXR analysis on all those samples. One of the features is that if some files already processed, let's say by your team member, then for the other one, if you use the same files, the results will be reconstructed immediately. That's what you see here. So the actual analysis of this data should take like 1 hour, more or less, on the scale. But since I already processed it yesterday when preparing to this webinar, the results appeared immediately. But in normal case, you will see the progress, etc. What's important here? Garbage in, garbage out. So the quality controls that are reported here, like alignment, tells us everything about the quality, the sequencing quality, and the library preparation quality that we have here. The green means the proportion of sequencing reads from the FASTQ file that was successfully mapped to the alpaca reference built in MyXR, and the yellow and red that are not. If we click on the individual sample, we will see this in the details. We can also go to the quality checks, where we have rigorous quality checks signaling that there are some alerts or warnings. We can expand and read the biological explanation. What's the potential reason of these, that, and that? And well, when we read, we can make our opinion. It's okay, we can proceed downstream, or we should like resequence, do something, or just remove and exclude the sample from the consideration. Yeah. For the bioinformatics folks, the logs of the execution are also available under the hood of each block, so it's fully transparent. Actually, the workflow of the block under the hood runs the well-known bioinformatics pipelines. And yeah, so with this, we annotated our raw sequences. We can look on them right away before moving to the next step. And to look on the antibodies, we can use Clonotype browser. That's the standard thing that we're doing. We're just adding Clonotype browser to see what we have there in the data. So I'm selecting the data set, and once the data loads, we see the actual results generated by MyXR annotation, by MyXR clonotyping. And by the way, here, MyXR clonotyping does not only annotation and alignment, it does error correction, which is extremely important. It does PCR and sequencing error correction using published and sophisticated algorithms that ensure that the clonotypes assembled by MyXR are error-free, confident clones. And in the clonotype browser, we can see our clonotypes, our antibodies in different samples that we have, or we can go to the overlap and see all of the antibodies from all of the samples that we have in the project right away here. And in this specific study, we don't have much, but we have something. We have 243,000 of unique antibodies through all of the libraries, through all of the samples. And of course, for every clonotype, we have all the information like the abundance, the CDRs, the frameworks, mutations, somatic mutations, and the CDRs, frameworks, etc., etc. All the information that we need for our down selection. Before doing the down selection, the standard step is to characterize our repertoire and see what we have, whether we have in general working phage and the funneling, the phage selection. Two components here. The first one, we need to understand whether the quality of the screening library is good enough. What I mean by this is that the aim of the screening library is to make it as diverse as possible, to have as much as possible screening candidates before we start phage display. And to assess this, we're adding the rarefaction analysis block. That's the instrument that once we select the settings and run, will let us understand whether we did enough sequencing of the screening and pinning libraries to get all of the variants, all of the antibodies that are there, and be confident that we're not losing something. Yeah. So it's just calculated. It's really fast. And let me just change some visualization parameters. So we have, just to remind, we have two samples from the screening library. Those are two replicates. Yeah, and two samples from pinning round 1 and pinning round 2, which are called here C1 and C2. So let me go to the configuration of the visualization. By the way, here, I will not go in deep into the explanation how you can play around with the visualizations, plotting, etc. functionalities. I'm focusing on the biology. But here, what you see, this graph maker is kind of prism graph pad that we have inside Platforma, allowing to build really rich publication-ready plots. So let me regroup the data here with enrichment round. So, and change some colors, just prefer the other ones. And also change the shapes. So for the interpolation, I want to use these ones. So good. Now what we see here, the interpolation means the dots from the actual samples. This rarefaction curves on the x-axis shows the sequencing depths, and on the y, the number of reads in the FASTQ file. And on the y-axis, it shows the projected estimated number of unique clonotypes or antibodies in our case that we get if we sequence to these depths. So for the screening library, the C0 round, that's the sample that we have here. You see it, yeah. We don't see the plateau. We see that if we increase the sequencing depths, we will still capture more clones, which is good. That's what we expect. The diversity of the screening library is our goal. If it is diverse enough, like ideally, it should be as diverse, so we are unable to sequence to the depths required to get everything there. Yeah, so it's fine. And we will check whether it's fine enough or not later. And for our pinning rounds, we see that there is a plateau. Yeah, for round 1 and round 2, the libraries, they are going to the constant, meaning that our sequencing depth is definitely enough, and we capture all of the antibodies that are present in the library. So this is a very useful type of analysis and QC to see how good we are with our libraries. The next thing to go deeper and validate this further is to assess the diversity of our libraries, diversity of our antibodies. And that can be done with the repertoire diversity block. So we select the data set, we run it, this block will basically calculate just a couple of diversity metrics with proper statistics, with a proper normalization. Everything is very, very correct. Let's go directly to the diversity graph. And let me change the visualization. Let's group our samples by the enrichment round. Yeah, that's what we have in our metadata. Yeah, let me also change the type of the visualization. And on the y-axis, let's first focus on the chow-1. So when we are looking on the diversity, we need to understand there are two angles of diversity. One angle is the richness. Richness is what we, for example, can estimate with chow-1 estimator. Chow-1 estimator is a diversity metric which predicts how many unique antibodies we would have in total in the library if we sequence to the saturation, to the full possible sequencing depth. And first of all, what we see here, we see a funneling trend from the screening library to the first round and second round. We see a clear selection. We clearly see a funneling effect. That's good. That means that our selection works. What else we see? That chow-1 estimator estimates that the total number of the potential number, the potential theoretical potential diversity of the screening library is around 180,000 of unique antibodies. If we get back to the rarefaction, what we have here, to which depth we sequence, that the last point, we sequence to the 160. So we are like under sequencing and losing around 20,000 or around 10% of the total diversity. And this is totally fine. So the problem is when we see like 50%. 50% is the problem. But here we are actually good. So the data is really good. Yeah, and we see the funnel. What else we can see here, and we always recommend to see, is one angle of the diversity is the richness, the diversity, and the other angle is the dominance. Whether this selection is derived by the fact that some of the antibodies are expanding, some of the clones are expanding, or just a random. And this can be estimated with, for example, Gini index. Yeah, Gini index is a standard metric for the dominance in the diversity matrix. And with Gini index, we see a clear signal that we are not just, losing the diversity because we are getting less of the antibodies, but we have a real expansion. The dominant, the diversity of the last round is dominated by like a couple of big expanded candidates, which is exactly the trend that we're looking for. So that's another validation of our selection. And the last validation that we also want to see is what's the path of this funneling, what's the standard path of this funneling, which we can easily see on the gene usage plot. So I'm adding the gene usage block, which will show us which of the V and J genes are actually enriching or not enriching or depleting from the data. So let's select the data set and run it. So it should be also pretty fast. It's a live demo. So I'm running the real analysis and doing everything in real, no fake. Let's also group our samples by, for example, enrichment round. Yeah. Let's do this just to make it nicer. And what we see here, so here we see the following. On the x-axis, we see our samples grouped by the enrichment round from the screening library to round 1 and 2. And on the y-axis, we have the V genes. And the color of the dot corresponds to the total abundance of this specific V gene in this sample. Yeah. So what we see here, we see that this now very much depends on what kind of antibody discovery you're doing and what you're looking for. But here we don't see like one clear winner, one clear V gene which is winning. we see a clear signal that some of the V genes are depleting. So these genes are gone. This one, yes, it's enriched, but not like dramatically enriched. This one is definitely enriched, but it was already enriched, so there was some bias in the screening library, yeah, which is fine for the natural source. It's alpaca, that's real immunized animal, and we always will have the distribution like this for non-synthetic, non-artificial libraries. So basically, here, what this picture tells us that once we select, the selection process is not biased to one, largely biased to one specific genes. It's definitely biased to a group of genes, depleting some of the combinations, which is pretty good signal for us is we aimed to select a diverse set of lead candidates. If we are aimed to select diverse set of lead candidates, then we'll have like the diverse set of genes enriched. Mark, by the way, for this picture is typical for animals, yeah, for mammalian species. This kind of distribution, yeah.
Well, yeah, of course. So like there's always some of the V genes will always dominate. And also it's
Why?
well, it depends, for example, for what tissue you work with. Some V genes,
PBMC.
yeah, so like for PBMC, like some V genes are more like you find clones with a certain V gene overrepresented, then you go to a different tissue and other V genes might be overrepresented there.
Okay. That's the characterization of our repertoire. Thank you, Mark. That's 3 blocks we did: verification, diversity, and gene usage to understand in general the repertoire, the NGS repertoire structure that we have and assess the quality of our selection. Now let's go to the down selecting the leads and families of leads. The first step here is that like why we're doing GS? We're doing GS because one of the reasons that Monica mentioned, let me just get back to the slide. One of the reasons that Monica mentioned that we want to find not just the singletons but the families of antibodies that will help us to find like more candidates if, for example, our lead fails, yeah, etc. And to do this, we need to group our antibodies in clusters, in groups. That we can easily done with the clustering analysis. We have a clonotype clustering block, which is based, by the way, on a very well-known bioinformatic tool called MMSCQS2. Let's add this block into the project. And see what we have. So let's select the data set and the parameters of our clustering. How do we want to group our antibodies into families? This depends on the biology that we are doing. For our study here, we are aimed to group antibodies with more or less identical sequences, yeah, protein sequences. That's why I'm selecting amino acid here. Yeah, by sequence, I mean full VDJ sequence. That's what is covered in our library. So I'm selecting here the VDJ region, the VDJ region in frame. Yeah, amino acid sequence. But if we're doing a synthetic library with like artificially constructed or mutated CDRs or frameworks, etc., then we can actually select the combinations here. So it's all possible. But in case of our study, we're using VDG region. That's what was sequenced, and that's where we are looking for the similarity. The alignment score is also important thing to consider. And again, it depends on the type of the in-vitro discovery that we are doing. We may select between exact match, which will just compare the number of identical residuals in the VDJ sequences of our antibodies, or Blossom similarity score. And Blossom is actually the alignment score, which takes into account the fact that certain proteins may have similar biochemical properties and certain proteins are very, very different. So the change of protein depends on what the protein is. So it calculates the alignment based on this very well-known Blossom matrix, and that's what we are actually aimed here for this study. The identity tells us how much of the similarity between two antibodies we expect to put them into the same group, into the same cluster. Depending on how we cluster, this should be a different number. Let's say if our minimal identity 95% on the full VDJ region in frame, which is, let's say, 100, well, less than 100 but more or less, amino acid lengths, then 5% dissimilarity means like roughly we allow 5 amino acid mutations. So that's how we can think about this. And we can loosen this parameter if we want to have larger families, yeah, or can increase if we want like a more strict threshold. And the coverage threshold is how much of the length difference we allow between this antibodies. So let's run it. This one clustering is actually a sophisticated algorithm. It runs under the hood the MMCQS with all the parameters, and we can see the logs. There are memory and CPU restrictions here, so it uses quite a lot of calculations. That's why we are waiting it here, because we are running live. And some people use CD heat for clustering, and I think the authors of this publication, they used exactly CD heat algorithm, but working with NGS data and on the scale of NGS data, MMCQS is the best option. So 1, 2, 1, 2, 3. Still should be pretty fast. Well, let me switch to the place where I already processed this, just to show how it will look like while it's running there. It's running, still running. So that's actually what once it runs, that what you will see. I'm just showing while we are waiting. It's running there. We'll see a table of our clusters, where each cluster is a group of antibodies with the similarity that we defined in the settings. Yeah, this block will calculate the centroid sequence for each of the cluster. The centroid is like the consensus, the antibody sequence closest to the theoretical consensus of this cluster. And then we can explore the content of the cluster on the multiple sequence alignment to see what's the content there. Where are those differences in the proteins allocated? And we can change this to, for example, CDRs and frameworks view to see where exactly the differences are allocated, etc. And this clustering information, so let me see whether, yeah, so I'm switching back to the project where I already, where we're all calculating, yeah. So, and this clusters now what we will use for the most important part of in vitro down selection, the enrichment analysis. So we have clusters of families of antibodies based on their sequence similarities. By the way, what we can also check, we can check how big our clusters are on this bubble plot, whether we have some like big clusters in terms of abundance and number of antibodies inside the cluster on this bubble plot visualization. And now moving forward to the most important part of our down selection, the enrichment analysis. Enrichment analysis is the statistical instrument that we are using to find families of antibodies that are expanding, that are selected with the selection. So let's use so here we have two options. When we look at the settings, two options. One option is number of reads. That's the number of reads of our antibody in sample, the abundance metric, or number of reads in clusters, because we can do enrichment analysis either on individual clone types or on clusters. And this depends on your in vitro antibody discovery setting. We use, and normally always recommend to use the number of reads in cluster, the condition column from our metadata as enrichment round, And the order of our rounds is from C1 to C0, sorry, screening library, C1, C2. There are a couple other options how we are doing this enrichment analysis, like downsampling. That's the normalization, since we are comparing abundances, and as we know, we sequence to different depths, meaning that the number of reads will be very, very different, and we need to have a normalization. So this normalization is performed here. And what else important? Since any enrichment analysis is very, very sensitive to the low abundant clusters or clonotypes, to the entities with zero abundance, then we need to configure how we deal with this. And I'm using shared clonotypes, meaning that since we're doing analysis on clusters, and we did a pretty wide configuration of the clustering parameters, we want to track only the clusters that are present in all rounds. So we are running this to get our, let's go directly to the frequency line plot and see that we have a clear window. So we have clusters that are clearly enriched, and the enrichment rate, the log-to-fold change, and the score of the enrichment is really, really high, which is awesome. That's exactly what we're looking for. And we can check what are those clusters and how the clonotypes from those clusters are represented. To do this, there are a couple of ways. One way is to use the clonotype space, the instrument that we have to just bring all of the antibodies on the two-dimensional plot for ease of like visual discovery. Let me select the data set, run this clonotype space block. And so here that's a UMAP plot where each dot is one antibody from the project. And the idea of the UMAP visualization is that two dots are located closer to each other if they share similar properties. And those similarity is measured by the protein sequence of the antibody. Let's go to the data mapping and use the enrichment score generated by the clonotype enrichment as a color to see that, like, the dark means the high enrichment. So those dark dots here are those antibodies that belong to really, really, really enriched clusters. And we can go deeper into them. We can select them. We can look at the content, what's the motifs there, and do a lot of discovery here. So a lot of things can be done here. And we will actually do this. And before that, let's do two more things. So if we have assay data for some of the candidates that we already tested, we can import this data and help us navigate the further down selection. So for this specific study, the nanobody is already reported. We know it. So, and there are functional properties from this nanobody. So let's use the immunoassay data log to integrate this information, the functional information, and match this information to our NGS data. So let me just move this block on top of the clonotype space. So select the data set. How we are going to match our assay data with NGS. It's super straightforward. We can import the assay data either from the FASTA file, or Excel file, or CSV file, where we have Sanger sequences and functional properties. And I have assay data file. I'm going to import. So from the assay, I have a CSV file with the functional information. I have a sequence column and other columns from this assay data. The most important for me is the target. So it's just the column which says that this Sanger sequence is the one that we picked from the functional screening. So the other parameters are same as for clustering, how we are matching the Sanger sequences to our NGS sequences. Let me put some strict parameters here just to do more or less exact match of our Sanger sequences to them. Yeah, so that's it. So what we have here, this is our Sanger sequence. There are some other functional properties that I'm not imported here, but just one important one, the marker, the target, SARS-CoV-2, the coronavirus. So that's the TY1 nanobody reported in the publication. And as we see in our NGS data, there are 41 antibodies that match this sequence with some like tiny allowance of fuzzy matching. So I allowed some 2% of dissimilarity based on the BLOSUM matrix to mark just more clones rather than like strict protein match. And you will see why. And if we now use our clonotype space, we will see the target column that we imported in the immunoassay data, and we can, let's say, highlight the ones that are from our functional assay data and show the enrichment. And what we see immediately, we see that the ones that are confirmed with a functional screening are really dark. They are the ones that are enriched. So that's already a good hit for us, and that means that we really have validation already with NGS just from this plot. But we can go further. The last step is the last two steps. One step is the developability. So for that, let's add a part of our standard pipeline, which is antibody sequence liability block, which will take the sequences of antibodies and calculate the standard liabilities that we know may damage the further, let's say, manufacturing or the further production of the antibody. There are different patterns that we can use. We can combine them. We can turn some of them off, etc., etc. So there are possibilities of customization. Why we're doing this? Because at the end, we want to see whether the selected leads that we have those liabilities or just liability, and then potential problems of developability or not. And then the last point is the antibody lead selection. That's the final place where we are aiming within the last few minutes to get our best candidates. So this is the block which will automatically pick the top hits, the diverse panel of antibody hits based on all the information that we have in the project. So first of all, it's the enrichment. And then let's say we want to pick like 50 candidates. Yeah, and we want to pick candidates which are first enriched, and second, let's say, most abundant. Yeah, have high abundance supported by many NGS sequencing reads. And we can configure this, and we can, depending on our antibody project, we can do this in different ways. We can find those that are most mutant, yeah, and have the highest rate of somatic mutations, or the ones that have like the largest distance to the centroid of the cluster, etc., etc. So here are our 50 candidates. First of all, let's see whether we have any candidates that match TY1 reported in the study. How we can do this? We integrated our target from assay data, and bingo, we have it. So we have, so you see this one, yeah. This one is the antibody, which is exactly the TY1, and it's confirmed by our functional data. But apart from it, we have many others, which if we sort, by the way, by the maximal cluster enrichment, yeah, this one is the most enriched. Our TY1 is the most enriched. But we see that there are others that are also very well enriched other candidates. Yeah, and that's means that in NGS we have a couple other candidates that we can then compare, see on the multiple alignment, and test further. So we're getting more. Monica, I know I'm running out of time. But this gives a pretty straightforward idea of how we can streamline the whole pipeline, make this as an automation, and with this, let's launch the third poll of our webinar.
Okay, great. So based on the workflow we showed today, how can we go beyond, and what advanced capability would you want to see come next?
Yeah, and yeah, let me go to the last slide of our
Perfect. So before we take any questions, so I just want to reiterate, so for how to access Platforma, we are completely free for academia and nonprofits. This has always been part of our mission to democratize access to tools. So all you have to do is go to platforma.bio and download it for free. For industry, we are happy to give you a free trial, and you can try out Platforma with your own data. All you have to do is book a meeting with us. I will drop the link below, and also send it out after this call. We have, as I mentioned, a special offer for attendees today. So if you book your meeting with us by the end of this year, you'll get your first 3 months free for your Platforma license. And because, as Sam mentioned, we have lots of people who are experts in this type of space, you'll be able to have a dedicated consultation session with us on your workflows. Okay, so I see the poll results are coming in, and let me just drop the link in the chat so you can book a meeting with us. And let's see. So it looks like we are tied for generative AI and also for 3D structure integration. And also, just looking at it now for complex formats, so specialized analysis for bispecifics, CAR-T, or non-standard scaffolds. So pretty split across the board here.
Yeah, well, the good thing here is that we already have a specialized analysis for all this other types of non-standard scaffolds that's already available, and the 3D structures is what we what we already have in the beta version. And AI, yeah, it's a trending thing. Monica, do we want to stay for questions? Or
Yes.
It's already like hard stop.
Absolutely. So we're right at the top of the hour. Feel free to stick around if you have any more questions. Otherwise, all our team will be here for the next couple of minutes. Otherwise, feel free to jump off.
Still sharing my screen, yeah?
Yes, you are still sharing your screen. Okay, well, that concludes today's session. Oh, we just got 2 questions. Yes, I will share with you the recording of the seminar, and Mikola has a lot of questions. We'll just do the first one for now, and then let's have a follow-up session with you. So you mentioned the tool is open source. Is it possible to contribute models and features for analyses that you do not have yet? If yet, yes, what is the channel for that and acceptable languages? So that's a perfect question. I think, Stan, you could introduce our SDK.
Sure. Actually, all of the blocks that we have, they are open source. Like every single block that we have on the platform is open source. So there is a link on the GitHub to the repository with the source code of the block, and we have some blocks. Well, not for antibody discovery, but for example, for the PCR specificity prediction developed by other groups, other peoples, or other companies. Yeah, we have an SDK in the platform and a full documentation on the SDK, how to build those blocks, those applications, and how to distribute them through Platforma. The languages that we use, obviously, so we provide this SDK to make it as easy as possible for bioinformatic developers. It's like all the blocks that we have, they're developed by our bioinformatic developers. The languages are for the user interface, obviously, like TypeScript, nothing else can be done. That's a user interface. For the workflow, we have the standard workflow language. Yeah, and that's it. There is a certain level of entrance, but then once you get it, you can build so many applications and expand the audience of users. So, and we also have a dedicated Slack channel for the Platforma developers, actually. So please reach us after the webinar and we can add you to the Slack channel.
Okay, great. Yep. How about we answer the rest of the questions offline, and we can